All right, why don't we get started with introductions as more people are joining the webinar. So for those of you who don't know me, I'm Kristen Schuler. I'm a wildlife disease ecologist at the Cornell Wildlife Health Lab, and I help with the New York State Wildlife Health Program. Kevin, do you want to introduce yourself? You have to unmute yourself first, though. I see your mustache moving, which might mean you're talking.
All right, I think I'm unmuted now. Hi, everybody. I'm Kevin Hines. I'm the Wildlife Health Unit leader of DEC. That's all I do.
All right. Next, we have Gavin.
Hi, everyone. Can you see me? I guess
Yep.
so. I am an anatomic pathologist, and I work in Long Island at the Cornell Duck Research Lab and do wildlife pathology down here and assist with everything up there whenever I can.
Great. Melissa?
Morning, everyone. I'm a research aide here at Cornell with the Wildlife Health Lab.
And Rachel?
Yeah, I joined the Cornell Wildlife Health Lab last fall. I'm a vet, came here from the National Wildlife Health Lab.
All right. And Lauren?
I'm bringing up the rear.
Saving the best for last.
Thank you. I'm a Fish and Wildlife Technician 1 with DEC at the Wildlife Health Unit.
Great, thank you. So what we're going to do today is to talk about emerging wildlife diseases. So I'm going to start off with a little PowerPoint about why we do wildlife surveillance and why it's important to you as a biologist. And then we'll go through some of the diseases that are kind of on our radar and why we're thinking about those. So if you have questions, unfortunately we can't do them live, but what we'd like you to do is type them into the Q&A box so we can capture them, and then I will read them to the presenter after the presentation and they can go ahead and answer them. And this is the third wildlife health webinar that we've done in this series, and the other two are available on the Cornell Wildlife Health Lab website. And this one should be available. We've actually been getting them up there pretty quick, so in the next couple days. So any of your coworkers that weren't able to make it, or if you have to step away for a little bit, they will be available there. So let's go ahead and get started. So talking about wildlife surveillance, I really like this cartoon because it really indicates how important it is to think about these new emerging diseases. And unfortunately for New York, you've seen more than your fair share of them in recent years, you know, with West Nile virus and white nose syndrome. So we really need to make sure that as people in the field that you understand why wildlife health is important and what we're doing for disease surveillance. So just kind of a primer of why biologists should care about disease. You know, we're always thinking about, you know, the population level. And when we think about populations, we think about recruitment, immigration, emigration, and mortality. So disease, we think, really has an impact on mortality primarily, but it can also impact the recruitment. And what we're seeing now is that disease is a natural part of the ecosystem for a lot of situations, but the impacts of disease are more devastating now because of what we're seeing with human activities. So whether it's landscape modification or people moving pathogens around the globe, those are really important changes that we're making to the ecosystem. And unlike with domestic animals where they have been able to eradicate different diseases, in free-ranging wildlife, we don't have many success stories like that. So we don't have the same tools in our toolbox with vaccination and depopulation that they can use in livestock and other domestic animals. So it's sort of one of those Pandora box type things where once the disease is out of the bag, we don't have a good way to put it back in. And actually what we're seeing now is a resurgence in wildlife diseases since the 1980s. So not only have we not eradicated disease, but we're seeing more disease happening. And we know that biologists care about these animals and so are thus in the best position to be informed. But unfortunately, that also means they're in a good position to be involved in the transmission. And as we've learned very succinctly with COVID these days, the transmission of zoonotic diseases goes both ways. So it can jump from wildlife to humans and then from humans back into wildlife. And we've recently heard about in the news a case where mink in Utah have been diagnosed with COVID-19, which undoubtedly they acquired from one of their human workers. The other reason biologists should care about disease and wildlife health is because it really does impact your ability to do your job. So this picture was taken at a training that I did in Oregon, where everybody was very worried about highly pathogenic avian influenza. And the public health officials really were talking about biologists having to go out in the field wearing full Tyvek, wearing respirators. And you can imagine how hot and uncomfortable this equipment would be if you had to wear this all the time because people were worried about what you might catch in the field. Therefore, it's really important for us to know what might be out there and to know what might be on the horizon to make sure that we're taking appropriate precautions to not only protect you from the disease, but also protect you from the health hazards that may occur from wearing PPE that might not be completely necessary. So we're always trying to balance those different risks. So I mentioned emerging disease already. And when we talk about emerging and re-emerging disease, that's considered relative to the populations that are being affected, where those populations occur, and then the magnitude of the impact and if it's increasing disproportionately. We also talk a lot about epizootic versus enzootic diseases. So epizootic would be something that we haven't seen before and that's increasing rapidly at a fast rate. Enzootic would be something that's existed in that area for a longer period of time. And one of the things that has come out from analysis of different disease hotspots is that zoonotic diseases are twice as likely to be associated with emerging diseases, just like we're seeing with COVID now, that it's a zoonotic disease, with a newly emerging disease is the non-zoonotic. So one of the ways that this can occur is through vectors or indirect contact, like we see with mosquitoes and tick-borne diseases. So all those sort of Lyme disease, Powassan, those are also emerging zoonotic diseases. So when we talk about wildlife surveillance, the World Organization for Animal Health classifies it as a systematic, ongoing collection, collation, and analysis of information related to animal health and the timely dissemination of information to those who need to know so action can be taken. So that's really the difference that I see when we start talking about surveillance versus monitoring. Surveillance, you are planning to take action. With monitoring, you're just kind of keeping track of what the disease prevalence is on the landscape. But with surveillance, like if we have an outbreak of chronic wasting disease, the very first case is going to trigger a lot of action. So why do we do wildlife disease surveillance? Well, the World Organization for Animal Health specifies the first two. So we want to reduce the cost to society from pathogens in wild animals. And these are actually not just the animals itself, but we have to think about the economic costs, the ecological costs, and then the social costs of these animals. We have an obligation to detect and report pathogens in wild animals because we are seeing these increasing number of emerging zoonotic diseases. And then I've actually added this last one because I think this is really important for people like us and where we're coming from. We need to ensure, because of our job responsibilities, that we have healthy wildlife populations through the early detection and management of these diseases. So some of the key points of surveillance is that it's a continuous activity. We need to make sure that we're prepared and remain vigilant for disease outbreaks. We need to look at the data regularly to make sure that we can detect any trends that might be occurring, this very sort of subtle patterns. We need to make sure that the people who need to know are getting the results of this data collection, that it's not just sitting in a file cabinet somewhere, and that we're distributing it to other agencies and people that need it, which is why we partner with the Department of Health and the Department of Agriculture and Markets so that we can inform our partners about disease events. So there's two major forms of surveillance. The first one is I termed passive. It can also be called general or scanning surveillance, which is what we primarily do. When you find a dead animal and send the carcass to Del Mar, we're doing passive surveillance that way. So that allows us to know which pathogens exist in New York and remain vigilant for emerging diseases. We have very few active or targeted surveillance programs. Chronic wasting disease is one of them, where we're looking specifically for that disease in a specific species. And we have well-defined sample sizes that we want from particular areas. And this is based on probability sampling so that we can know if the disease is there or we have a reasonable assurance that it's not there. So this allows us to plan how the animals are going to be collected and tested in advance. So when we do disease outbreak investigation, we want to make sure that people are prepared. And unfortunately, we couldn't go out this year and do the workshops that we normally do. Here's a picture from training down in Region 1 that we did a few years ago. But having the equipment and knowing what you should do in these situations is really important because we don't have a standard definition for an outbreak. And a lot of that is because we don't have the baseline data. We don't know what normal mortality should be for every single species out there in every location. We also need to think about what an outbreak might do to human and livestock health. So we're not just thinking about the wildlife in this context because a lot of these animals are going to occur on private lands, and then try to figure out what the potential impact is on that wildlife population. Is this a unique occurrence? You know, was there some freak weather event that killed a bunch of animals, or is it something that is the first case of a new disease that we've never seen before? So just to wrap this up, we need to keep in mind that there is potential for zoonotic transmission between wildlife and humans, and making sure that you keep that in your head is critical for biologists in the field. And one of the things that I've come across in my career is that when you go to your doctor and you say, you know, I work with wildlife, they tend to say, well, that's great, you know, but they don't necessarily think about what you might be exposed to. And I know a lot of you already have these wallet cards. We pass them out at the other workshops, but this is just a card that you can take with you that gives you a little bit more credibility if you're worried about something that you may be exposed to in the course of your job. And it kind of just makes that doctor think more broadly about what you might have been exposed to. So you can download these yourself from this website and print them out and keep it in your wallet. So it's a nice thing to have. So let's see, I saw a question. All right, so one of the questions was, any thought as to why the resurgence of disease started in the 1980s? That's a really good question. I'm not sure that I have a great answer for that. I think one of the things is that we did start seeing, you know, increased human populations. We started seeing more global travel, more globalization with trade. So people moving more things around, you know, people moving into the suburbs. So it's just the movement of animals and people really can spread disease quite a bit. So that would be my answer, but I, again, would welcome other input on that. All right, so that's what I have. I'm going to turn it over to Kevin now to talk about rabbit hemorrhagic disease virus and EHD.
So good morning, everyone. This morning I'm going to talk about rabbit hemorrhagic disease virus 2, which is a viral disease of rabbits that is currently, we're currently experiencing an outbreak in the southwestern part of North America. And we're certainly concerned that this disease might come to New York and cause problems for both our domestic rabbits here and our wild rabbits. And at the end, I'm going to say a few words about epizootic hemorrhagic disease, which is another hemorrhagic viral disease. This one affects deer. And the reason we're going to talk about that today briefly is we're getting into the season where we're likely to see some cases if we're going to get any cases in New York this year. So rabbit hemorrhagic disease was first identified in China in 1984 in some domestic rabbits that were imported from Germany. And this virus quickly spread through the domestic rabbit populations worldwide. And it has a very high mortality rate, sometimes up to 100% in certain populations of domestic rabbits. It's easily spread by indirect or direct contact. The rabbits get it, nasally, orally, through mucous membranes. And the virus is very stable in the environment, which I'll talk a little bit about in a couple minutes. Another strain, rabbit hemorrhagic disease virus 2, was first discovered or identified in France in 2010 in domestic rabbits. This other strain of rabbit hemorrhagic disease has the ability to go into wild rabbits as well. So the original strain of rabbit hemorrhagic disease virus just affected domestic rabbits, and it didn't go into wild rabbits. But this new strain has the ability to go into wild rabbits, which makes it a little more vexing for us. And this map over on the right shows the occurrence of rabbit hemorrhagic disease virus 2 over the past 10 years or so. And you'll notice that in the southeast or southwest US, there's a lot of red dots currently. So this rabbit hemorrhagic disease virus 2 virus is very hardy, and it can remain viable in decaying carcasses or on surfaces, in bedding, on caging, for 3 to 4 months. When I say remain viable, another rabbit can come and have contact with it, and they can become infected over a period of 3 to 4 months before the virus is no longer infectious. In frozen rabbit meat, the virus can last at least 3 or 4 months and probably indefinitely. And some folks suspect that frozen rabbit meat or pelts or rabbit products, the virus can also be on the hair because the rabbits are grooming themselves. These are ways that they suspect that this virus has been moving around the world and around the country. As I mentioned before, this strain of the virus, rabbit hemorrhagic disease virus 2, can infect wild rabbits. And we know from experiments done at Plum Island Animal Disease Facility a couple of years ago, they exposed eastern cottontails, which is our most numerous native rabbit in New York, to this disease orally and nasally. And they exposed 5 rabbits, and 3 of them died within several days. So we know that our wild rabbit is, the eastern cottontail, is susceptible. We have no reason to believe that the New England cottontail isn't susceptible, and we have no reason to believe that the snowshoe hare won't be susceptible as well. So I think all 3 of our native rabbit species are going to be susceptible. In wild rabbits, typically there's no symptoms or signs. You typically just find the rabbit dead. And it's a hemorrhagic disease, so one of the few signs you would see on the outside of the rabbit might be blood from the nares or nose, like you see in this photograph on the right. You also might see some blood coming from the anus or the vulva. When we open up the rabbits in the lab, we might see some disseminated hemorrhage in any of the organs with this disease, in the lungs, or even free blood in the body cavity. We'd also expect to see some liver necrosis because this virus replicates in the liver. So we've had some periodic incursions of rabbit hemorrhagic disease virus and rabbit hemorrhagic disease virus 2 in North America over the years. Over the past couple of years, there started to be some outbreaks in British Columbia and Vancouver Island and Washington State. On Vancouver Island, there's a population of feral domestic rabbits, European rabbits, which I guess is quite large, and they had some outbreaks in that population. Earlier this year, in late April, March, there was an outbreak at an exotic veterinary facility in Manhattan, New York City. They were boarding some domestic rabbits. They had 13 rabbits, and 11 of the rabbits died in short order. USDA and New York State Ag and Markets investigated that outbreak, trace backs, trying to find, you know, where the initial infection came from, and they were unsuccessful in doing so. We know very little actually about how this virus is moving around the country. You know, we have certainly ideas of how we think it's happening, but we don't know for sure exactly how it's happening. So the outbreak at the veterinary facility in New York City is over. We didn't find any wild rabbits exposed. Luckily for us, there's not a large eastern cottontail population in downtown New York City and Manhattan, although they do occur there. Around the same time, they started to see outbreaks in New Mexico, Arizona, and Mexico and Texas in domestic and wild rabbits. And this was the rabbit hemorrhagic disease virus 2. It's a different strain than the strain that they found in New York City and in the Northwest. But this, which indicates that this was a separate incursion into North America. And currently, the virus is pretty widespread in domestic and wild rabbits in many of the states, most of the states of the Southwest. And they are seeing anywhere from, wildlife biologists are saying they're seeing anywhere from groups of 12 to as many as 100 rabbits dead in the field. So possible modes, you know, naturally we're concerned that this virus is going to make its way back into New York, and it's going to get into our wild rabbits. So possible modes of introduction would be movement of domestic rabbits, their cages, their bedding, their food or contaminated food, rabbit parts, meat, hides, all can carry the virus. And the virus can even travel on fomites. Fomites are like little pieces of soil or dust that the virus is attached to, and they can blow around in the wind that way. They get on your clothes. They can certainly get on your shoes. The virus can get on your shoes. And we're concerned maybe that someone could possibly go hiking in Arizona and step on some rabbit poop, and they could bring that back on their shoe and introduce it to New York. We really don't know how it's spreading, like I mentioned before. On the domestic rabbit side, rabbit shows and county fairs are areas where, or situations where rabbits are brought from different areas and congregated for a period of time. Folks that keep these domestic rabbits, they have rabbit shows just like people that are into dogs have dog shows and cat shows, etc. And then they travel from state to state with the rabbits. And county fairs, certainly 4-H situations, you've got a building full of rabbits that all the kids are showing the rabbits, and that is certainly a risk of moving that virus around. We've been a little bit lucky this year. I don't know if luck's the right word, but COVID-19 has kind of put a damper on a lot of county fairs and rabbit shows and a lot of movements. The virus also has been documented to move from biting insects from rabbit to rabbit, so fleas and ticks, and also flies and biting flies can transfer the virus. So the chances of it coming to New York, well, they're pretty good because we already had it here once, and we don't know how it got here, so we don't know how to stop it from coming again. This virus is resistant to some of the common disinfectants like Clorox and Lysol. Disinfectant wipes aren't really effective against it. Quaternary ammonium compounds, which is what we use routinely in our lab to disinfect the necropsy rooms, is not always effective against it. So when we have a suspected case, or if you have a suspected situation where you think this virus is present, one of the more effective things to use and easiest to get is 10% household bleach, or Virkon S is a common veterinary disinfectant that's available. As you know, 10% household bleach, it's 1 part fresh household bleach, 9 parts water. You want to mix that solution fresh every day. It deteriorates over time. And you want to have a 10-minute contact time for if you're going to use bleach. So to reiterate the potential avenues of introduction into New York, we're concerned about the movement of domestic rabbits, their bedding or caging into New York, the movement of their parts, meat, hides, pet food that might be made with materials that are from an area where rabbit hemorrhagic disease is occurring in the Southwest, and contaminated shoes and clothing. Certainly the movement of wild rabbit carcasses and parts would be a concern. If a hunter from New York went out and was hunting in New Mexico or Arizona and brought back some rabbit parts or material on their shoes, that could certainly be an avenue of introduction into our New York population as well. And wildlife rehabilitators who also keep domestic rabbits are a concern. We've reached out to them with some education materials suggesting that they don't have any contact if they routinely do a lot of wild rabbit rehabilitation, not to keep domestic rabbits, not to have contact with other people's domestic rabbits, and not to have volunteers that keep domestic rabbits helping them out in their facility. Dog trials and trap and transfer are another potential risk for moving virus around. This is a situation where they have rabbits in penned areas, and they use trained dogs to work these rabbits. And folks that do this can travel from state to state doing it. And it certainly leaves the possibility of having the dog's feet or other equipment contaminated with virus and then move them back to a different state. If we get rabbit hemorrhagic disease virus in our New York wild rabbits, as Kristen mentioned earlier, once Pandora is out of the box, it's hard to get it back in. It'll be virtually impossible for us to control this virus once it's into the wild population. There is a vaccine for domestic rabbits that currently you need to get special permission to get into the state to use. And they are going to, I believe there's a domestic manufacturer that's working on it, and they expect to have it more available for domestic rabbits in the US in a year or so. But there's no practical vaccine for wild rabbits. They'll have to come up with some kind of oral vaccine, would probably be our best bet, which certainly doesn't exist currently. We might expect to see population reductions as high as 70%, maybe even more. We're not really sure how it's going to play out. And this could be an issue because rabbits kind of form the base of the food pyramid for a lot of our predators and scavengers in New York. You've got eagles, red-tailed hawks, great horned owls, foxes, coyotes, fishers, timber rattlesnakes, all prey on eastern cottontail rabbits. And certainly they get hit on the roads a lot, and they provide food for vultures and crows, my favorite bird. And then there would also be less opportunity for rabbit hunters if there's fewer rabbits. And we're very concerned about how this might affect our efforts to conserve the New England cottontail populations in the eastern parts of the state. So now I'm going to talk about epizootic hemorrhagic disease. So what I didn't mention before, the rabbit hemorrhagic disease virus only affects rabbits. It doesn't affect people, it doesn't affect livestock. Epizootic hemorrhagic disease virus generally just affects cervids, although sometimes the virus can spill over into cattle, and the related blue tongue virus can certainly go into cattle, sheep, and goats. But neither of these viruses go into people. Epizootic hemorrhagic disease virus is spread by a biting fly. Down south, they call them no-see-ums. The known vector is Culicoides varipennis, but other species of Culicoides could also be vectors. It is the single largest viral-related cause of death for deer in North America. And this virus can come in and it can kill hundreds of deer in an area in a short period of time. This photo on the right are some deer that are kind of bloated in the water. And this is a somewhat typical situation to find deer that have died from epizootic hemorrhagic disease from. You've got multiple deer, you've got them in water, and they're already bloated by the time you find them. This virus was first identified in New Jersey and Michigan, both back in 1955. And outbreaks are localized and they occur in the late summer, early fall which is why we're talking about it now, because we're approaching the season where we might see this. This virus is endemic in the southern states. They have some of the deer have some immunity to it down there. In New York, certainly we don't see many outbreaks, and our deer are considered naive to this virus. So in my opinion, in most cases in New York, when they get infected with the virus, they're going to die from it. They're not going to have any immunity at all. In 2007, we had an outbreak in Albany and Rensselaer counties, and then we found, I believe, one deer in Niagara County that had it. Back then, I was allowed to go outside the building, out into the bush, and we went out and were beating the bushes. This outbreak actually occurred not too far from the lab initially. And we were able to find carcasses by watching the vultures, and the vultures kind of told us where the carcasses were. They were dead in the woods, and the vultures could find them. And we think there were like 50 to 60 cases in the Capital District related to that outbreak. And then we didn't have another outbreak again until 2011. In 2011, down the Clarkstown area, I think the way that they were alerted to it was the highway crews were seeing getting a lot of calls for roadkill deer that they didn't think were hit by cars. They were near the road, but they weren't necessarily hit by cars. And our field staff started to look into it, and it turned out there was cases of epizootic hemorrhagic disease, and they estimated that they had 100 or so mortalities related to that. So as I mentioned before, the carcasses are generally found near the water. We think that might be because they get feverish when they're infected with this virus. Their body temperature is elevated, and some folks think that maybe they're going to the water to cool off, and that seems plausible to me. And you also see this rapid deterioration of the carcass after death. Decomposition sets in a lot faster with these animals, and they bloat up really quickly. And that could potentially be because their body temperature is already elevated when they're dead. Also, a lot of the blood vessel walls and tissue walls have kind of deteriorated, and allows for quick movement of bacteria within the carcass. With live animals, you might see a rapid respiration rate. The head and tongue can become swollen. We saw some of that in New York. Down south, where the disease is endemic, some deer have some immunity to it, and they have this chronic form where they might be losing body condition. There's some hoof lesions, some laminar problems with the hoof growth. You see these lesions in the coronal area of the hoof. And you also might see some lesions in the mouth, up on a hard palate or on the tongue, some large open sores and lesions. This is a shot of one of the New York deer. You can see the tongue is swollen. There's a lot of blood in the fur, probably coming from the nose. This is another one where you can see that the bottom of the jaw is swollen, and you'll notice that the conjunctiva, the sclera, the white part of the eye, is hemorrhagic in this case. And these are the kind of signs that we saw back then. It's important in these cases to try to get the deer into the lab as quick as possible because they deteriorate so quickly. But we should be able to get a definitive diagnosis via PCR, even on some decayed carcasses. This is our lab with a rainbow over it. I took that picture. That's
excellent. Very nice picture, Kevin. All right. You have quite a few questions.
Oh, wonderful.
I think you might have answered this one already, but somebody was asking about the vaccine for RHDV, and then there was a question about the feasibility of giving the vaccine to New England cottontails prior to release.
Well, if you had the New England cottontails in hand and you had the vaccine, it would be feasible. It's not a long-acting vaccine. So as I understand it, you would need a booster every year. So that would buy them a year, maybe a year and a half of protection. And then if you weren't able to recapture them and revaccinate them, it would have potentially limited utility.
Great, thank you. Another question, is RHDV2 transmissible through urine? Would the use of urine for trapping or predator hunting be a possible source of transmission?
Absolutely. It's definitely transmissible through urine and feces. And that's also another potential problem for rabbits is they're coprophagic, which is kind of a cool word for means eats their own poop and probably eats the poop of other rabbits. So the virus is being the rabbit's urine and poop. And if another rabbit comes and eats the wrong poop pellet, they could get exposed that way, and certainly by urine as well.
All right, thank you. Another question, is alcohol, for instance, the New York disinfectant we've all become familiar with, an effective disinfectant for RHDV2?
That's a good question. I do not know. I would say no until proven otherwise, until I look it up and see if it, yeah, I really don't know.
Okay. Another question, any evidence that it affects snowshoe hares too?
Well, we've got no experimental evidence, but we know that down in the Southwest, they've got a number of hare species that are being affected. So there's really no reason to believe that our snowshoe hare or varying hare is it has any resistance. I'm sure it's susceptible. I would bet $10 on it.
10 whole dollars?
Yeah.
Wow. Okay. Another question, is there a lab test you use to confirm?
A rabbit hemorrhagic disease?
Mm-hmm. Is it diagnosed pathologically alone, or is there a lab test to confirm?
Oh, yeah, there's a confirmatory lab test. It doesn't grow well in tissue culture, so PCR is the best test to use. And currently, we've tested a couple of rabbits this year so far, and those are going through USDA to Plum Island to the Foreign Animal Disease Lab out there. National Wildlife Health Center and Southern Cooperative Wildlife Disease Project both can do testing for it in wild rabbits, and I expect that a lot of the veterinary labs, including yours here in Cornell, will have that test online in the very near future if it's not online already.
All right, I'm going to cut off the questions here because we've got to move on. Well, I'll ask you the two remaining questions. So what is DEC's plan for when RHDV2 comes to New York? Will there be any hunting restrictions? Will you be testing wild rabbits found deceased? Is that something you're already doing for surveillance?
So far, we've put the word out to field staff about this disease and to wildlife rehabilitators. So we're looking for rabbits that are sick or found dead. So we're trying to do surveillance that way. We currently don't have any restrictions on the movement of domestic rabbits into and within New York, which is something I would like to see happen because that's, in my opinion, is the greatest risk currently. As far as hunting restrictions, I'm not quite sure how we'll handle that. I guess it would depend on how the population fares. If it really wipes them out, there's not going to be many rabbits to hunt, so we'll probably put some restrictions in place to try to bring them back in certain areas.
For the remaining questions, we can type some answers into the Q&A section. So either Kevin or I can do that. So Kevin, if you want to stop sharing, we'll turn it over to Gavin.
Escape. This one.
Hello, everyone. Can you hear me?
Yep.
We're all seeing a single PowerPoint slide at a time, I hope. Excellent. I don't want to give away the good stuff. Okay, so I'm Gavin. I'm a pathologist down here in Long Island. I've been helping with the wildlife folks since I think 2015 or 16, doing some cases and sort of looking at what's going on in Long Island region. And then I read some biopsies from the rest of the state. So I get to see kind of some really cool cases. Today I'm going to talk about adenoviruses with a specific focus on cervid adenovirus 1 and canine adenovirus 1, and I'll do a quick little blurb on canine adenovirus 2. Starting off with cervid adenovirus 1, and then we'll go into the canine adenoviruses. So adenoviruses in general are icosahedral DNA viruses. Pretty much every species has their own adenovirus. Most of these viruses result in a subclinical or an aclinical infection with some notable example or exceptions to that rule, which I'm going to talk about in these three specific adenoviruses. Most often the disease is evident in young or immunocompromised animals, but again, there are some exceptions to that. Most of these adenoviruses are species specific or within a class of species. So the cervid adenoviruses stay within the cervids. The canine adenoviruses tend to stay in the carnivores, and they don't really mix back and forth. Sequencing of these viruses has sort of sorted that out pretty nicely. So that's one nice thing. We don't have to really worry about deer adenoviruses in coyotes. However, there are outbreaks in naive populations, and that's where we're going to sort of focus on the cervid adenovirus 1. So this is oticoleus adenovirus 1. Deer adenovirus 1 is how I'm going to refer to it. It's a little easier to get across. It was formerly known as cervine adenovirus 1. This is principally a Western state disease, California, Utah, Oregon, Arizona, that region, as opposed to anything we would see in New York. But we are monitoring for this, of course, at all times. So they first discovered this in 1993 and 1994 with some outbreaks. I believe it's in black-tailed deer and mule deer in California. And they had pretty significant mortality in those outbreaks there, with several hundred deer found dead. And those are brought to the CAS laboratory system in California, and they identified this virus. They worked out the transmission means in those outbreaks to be a direct contact with densely populated deer and possible vector-driven transmission, but I don't know that that's been confirmed at this time. So species affected with this are, of course, the cervids, specifically the blacktail and the mule deer tend to be most severely affected. But white-tailed deer, moose, pronghorns, and probably others are also on that list. Fortunately, we haven't really worked that all out, or unfortunately, we haven't really worked all that out yet. Clinically, these deer are going to present as weak. Might have open mouth breathing, labored breathing. You'll see them trying to force air in and out of their lungs. They might be drooling, and then they're often just found dead, either with bloody diarrhea or like this image here, with foam coming out of the mouth and nose in those areas. So as I was saying earlier, it's predominantly in the Western states, but it is moving eastward. As you start looking at cases, which I'll go into some of them, you start seeing more and more cases working their way east. So hopefully it sort of isolates itself to the West Coast and we can keep it trapped there, but we're always monitoring. So this is a high risk level for me, especially in naive populations, particularly young deer. Mortality rates can be quite high. It can wipe out crops of fawns pretty quick. It's not a zoonotic pathogen, meaning we don't need to worry about it, at least at this time, going from the deer into us. It doesn't mean we don't provide appropriate protective equipment, but at this point, they've shown transmission from a deer to a person. Differential diagnoses for this disease include epizootic hemorrhagic disease, as Kevin was talking about, and as well as blue tongue virus, an orbivirus that can cause similar gross lesions and microscopic changes. So gross pathology, main findings of pulmonary edema, and we'll see these hemorrhages throughout multiple organs, principally the liver, as well as the kidneys and potentially on the omentum, some of the serosal surfaces of the intestines. Another finding that they found in these outbreaks in California was pretty prominent abscesses in the mouth, tongue, throat, and along the foregut. And what that looks like is right here. So what we see in the top left image here from image A, you see the lungs have these sort of modeling red and white, and there's actually, if you look closely, increased space between the interlobular septa here, which is expanded, and that's all pulmonary edema. This deer would have froth coming from the mouth and along the entire length of the trachea, most likely. What you see on image B is the larynx and the cartilage and the surrounding pharyngeal tissues in this area has undergone necrosis from the vasculitis, and then a secondary bacterial infection sets up in here. So when you see this, it's probably not going to recover the virus from that lesion due to the necrosis and secondary bacterial disease, but that's sort of a giveaway that, hey, something happened to that tissue which resulted in the secondary opening of that for bacteria to come in. You see that again in image C down here. You see a nice necrotic tonsil, or excuse me, this would actually be, yeah, probably a pharyngeal lymphoid tissue, maybe a lymph node in that area is necrotic. It's probably been devascularized from the vasculitis, and as well as the tongue here, you'll see an erosion and ulcerative lesion in the tongue as well. You see again secondary bacterial disease filling up here with these more suppurative, nice cheesy foci in those areas. And then as you extend down into the stomach, this is actually a section of the rumen that's been opened up. And what you'll see here is these nice areas of dark red, which is indicative of vasculitis and necrosis secondary to devascularization of that tissue. You'll see a thrombus here. So histopathology, it'd be a shame for pathologists to not at least go over some of this, so I had to do it, so bear with me on this. Microscopically, we're going to see pulmonary edema, which I talked about, hemorrhages widespread throughout multiple organs, and then vasculitis with secondary necrosis to that tissue as it's devascularized and devitalized in nutrients and waste removal. And then intraendothelial, meaning inside the vessel walls, they have these nice intranuclear viral inclusion bodies, which I'll point out. So working through these images again from that first report in Davis, they show these nice long septa that are expanded by this fluid. This is pulmonary edema. And you'll notice that they're going to highlight this, some of these vessels here in this image here, which we'll come back to. They put in a nice section of intestine here, which has nice areas of hemorrhage and necrosis of the villi that line that mucosa. You can see that they're just sort of blown apart. The higher magnification here, you'll see that this is actually a vessel, and you'll see inflammatory cells moving through that vessel wall and destroying the lining of that. That's indicative of vasculitis. And then the higher inset here, they actually show one of these remaining endothelial cells containing an intranuclear viral inclusion body, which for a pathologist is kind of the yay moment. We actually know what's going on or have an idea of what's going on. Now we go to send it to PCR for confirmation. This is actually an immunohistochemical stain they developed, and you can see that it's lighting up these endothelial cells, further supportive of that adenovirus infection. So just some timely literature here is in a case in Arizona where they had mule deer. This is March 17th of this year, so it's moving east. And then again, it's sort of in the population in California. They had another outbreak here in August of this year as well. So control, no vaccines are developed at this time. Focus is trying to eliminate congregations, a large number of deer to minimize that spread between the deer. I kind of like this sign. I might have to get some for the laboratory property. But anyways, so don't feed deer, discourage people from doing that, and that will help slow the spread of this virus across the country. And now I'm going to switch gears a little bit, move into some carnivore adenoviruses, focusing on canine adenovirus 1 and canine adenovirus 2. Species affected by this are dogs, coyotes and wolves, fox, bears, lynx, and actually pinnipeds, including sea lions. I think some seals they actually recovered these viruses from, so that's kind of an interesting comparative pathology point. These are not zoonotic viruses, getting back again to that point that these are typically species-specific viruses, or at least within a class of carnivores, they're going to be able to share this as opposed to switching over to humans and vice versa. So I'm going to focus on canine adenovirus 1. We had some nice cases earlier this year in some fox and actually a coyote from New York. And in these cases, the clinical histories was more of an acute phase, which is what we tend to see in the wildlife where there's a rapid disease course. These animals presented with anorexia, lethargy, and depression of about a day, and then neurologic disease was setting in within 24 hours, and the animals were found dead, or the animal was just found dead in the case of the coyote that we had. These viruses, these canine adenovirus 1s have a similar tropism, meaning they like the vascular endothelial cells as well as hepatocytes in the liver. which is kind of the secondary name for this, which is infectious canine hepatitis. So that's sort of where it gets the namesake. There is a chronic phase, but it's not as well described in wildlife, where they can get an anterior uveitis, or what we call blue eye in dogs. And this is more of a delayed hypersensitivity reaction to the infection, they think. But again, it's sort of not as well described. Most often we see the acute phase in these animals. Transmission is typically direct contact with infected feces, urine, and saliva. And some reports have shown that they can shed for months, at least in dogs. So we assume similar shedding capacity in other animals if they do survive or have a transient infection, clear it, and then they might not die from it, but they can still shed into the environment. So that's sort of a risk factor for us with this canine adenovirus 1. So grossly, what we're going to see, if we see anything at all, is we might see hemorrhage in multiple organs. You might be lucky and actually pick out a necrotizing hepatitis, like you'll see in this image here, these black and red, slightly depressed areas on the liver surfaces. This is actually a canine herpes virus, just because I didn't get a good image of an adenovirus, but that's a good differential for any sort of hepatic necrosis in a canid species. And you might be lucky and see the gallbladder edema that they'll describe in some of these canine adenoviruses. But most often we find these animals just dead without anything, maybe a little bit of hemorrhage here or there, if you're lucky to get a fresh carcass. Microscopically, it looks similar to the deer adenovirus, where we get a systemic vasculitis. Often they have pulmonary edema. It'll look very similar. Hemorrhages throughout multiple organs. And then in the canine adenovirus 1, it tends to knock out the liver. So you'll see this, what we call central lobular necrosis, and the hepatocytes just start exploding and dying. And then they will die from liver failure very quickly. And then, of course, sort of the slam dunk for a pathologist here when we see this with that history is these nice intranuclear viral inclusion bodies here, and up there, often lining the endothelial cells around a vessel or in the sinusoids of these livers. So this is actually a case that I had of the coyote that Kevin, your group, did the gross necropsy on. This is a section of the brain of that coyote, and you'll see a vessel here. You see degenerate inflammatory cells sort of percolating through the wall, blowing up that endothelial cells, and you can see an endothelial cell here swollen by an intranuclear viral inclusion body. And then this is an immunohistochemical stain that we have in-house, and you can see that it lights up that cell very nicely. So we actually confirmed that case with virus isolation and PCR for canine adenovirus 1. So it was a nice case. Control of this, I don't believe they use this in the wildlife, but use of this in the canine populations has dramatically reduced canine adenovirus 1 with its cross-protective ability by using the canine adenovirus 2 vaccine in the United States. So this has sort of knocked down the amount of virus that's out in the environment, thus decreasing wildlife exposure. So we periodically will get pop-ups with these young, naive animals that are exposed from a maybe persistent shedding animal, but at least it's not spilling out from the domesticated animals into the wildlife population and back and forth feeding on each other. Just quickly, canine adenovirus 2. This is a similar adenovirus. This one is a little bit different, though, in the fact that it tends to target the respiratory epithelium. It's a member of the diseases that tend to make up what we call kennel cough, and is why we have that vaccine to help prevent this disease. And we're just fortunate that it gives a pseudo-immune response against canine adenovirus type 1. These are typically self-limiting infections unless you have a really young, probably immunocompromised or stressed animal. It can turn into a terminal necrotizing bronchitis and bronchiolitis, which is infection of the lower airways, often compromised due to secondary bacterial infections in those cases. And with that, I'll take some questions. Do we have any?
You do have questions. Somebody would like you to explain PCR.
Okay. I am not a molecular virologist or anything like that, but I will give it a go. So it's polymerase chain reaction. Basically, they extract a component of the virus, either the DNA or RNA, and then they break it down into the ATCG components. They get the processing and repeat section of that. They identify it, they amplify it, and then that sort of gives a blueprint for saying this is this virus versus some other virus. How'd I do?
All right.
Okay.
Yeah, so basically if the chunk of virus is there that they're trying to find, the PCR process amplifies it and then it can be detected. So if it's not there, it doesn't get amplified.
I always think of the Jurassic Park scene in the original one where they show that little nucleus or nucleic acid thing swirling around, and they're just basically making more of those to identify. That's how I remember PCR.
Pretty much, yes. All right, excellent. I will turn it over to Melissa now.
Good morning. I'm going to talk to you guys about B-cell, which is an emerging disease threat to our native salamanders. So what is B-cell? B-cell is Batrachochytrium salamandrivorans, also known as salamander chytrid disease. It's closely related to BD, or the chytrid that's caused global extinctions of frogs. So B-cell is a fungus that causes skin lesions and ulcerations, secondary bacterial infections associated with those. The animals become anorexic, develop neurologic signs, and typically die within a week of clinical symptoms. The pictures here, you can see the salamander is a fire salamander, and he has sort of a thickened skin sloughing. He has lesions, and then there's histopathology that shows the actual fungal spores within the epidermis. So where is B-cell currently? It was first seen in 2008. There was a wide die-off of wild fire salamanders in the Netherlands, and it was formally described in 2013 and has since spread to Belgium and Germany. It's also found in Asia. They think it has originated in Asia, and the salamanders there have a natural immunity to it. They think that they co-evolved and are able to carry and become diseased from it. So why is B-cell particularly concerning for us? So North America is the global salamander biodiversity hotspot, particularly the southeastern US. And so you can do risk assessments and vulnerability models, and our whole area is mapped to be highly vulnerable to B-cell outbreaks. And this is based on species richness, habitat concerns, as well as where the major ports are. So the map on the top right, you're looking at all the ports where salamanders are often imported. And the year that this was mapped, there was 779,000 salamanders imported, which is a lot of salamanders, 768,000 of which they determined to be a risk of B-cell because they either originated from Asia or passed through a port that was in Asia prior to coming to the US. So what is being done about it now? Fish and Wildlife in 2015 listed 201 species as injurious under the Lacey Act. So injurious can be either a threat to human health, or is typically used for forestry, agriculture, horticulture, anything that would be impactful of that, or also potentially detrimental to wildlife species, which is the case here. So under the listing, it includes our native Notothalamus. So newts seem to be particularly susceptible to this, as well as plethodontids, so many of the lungless salamanders that we have. Although when they did infection trials, we also have some animals like our ambostomatids, so spotted salamanders, marbled salamanders that can carry it, but don't necessarily become diseased from it. So there's a huge task force that includes state and federal agencies, NGOs, different research institutions, and they set up this framework for surveillance and monitoring, as well as response if it comes to the US. It should be noted that it's not currently found in the US, and actually, just as of last week, there was a new article published where they had tested 11,189 samples from 35 different states and did not find it in the US yet. So that's good. Although some people seem to think that it's imminent, unfortunately. So what can we do about it personally? First and foremost, biosecurity. I know that we sort of pound this into your heads and you're all familiar and I'm sure follow the protocols, but field site to field site, you want to be disinfecting all your equipment. And this goes for both work sites as well as recreational endeavors. So if you go fishing, anything in the water, you want to use either bleach or Vircon or something. And partners in reptile and amphibian conservation did a really good disinfectant protocol, which we'll link in the comments for you. So second, disease surveillance. So ideally, collection of any dead or dying salamanders so that we can do necropsy and test for B-cell. We do have a B-cell PCR available now, which is pretty cool, although hopefully we won't need to use it. So if you collect salamanders, they don't freeze well. I know we often request frozen tissues if you don't send them in, within 24 to 36 hours. But in this case, salamanders, any amphibians really, it's ideal to put them in ethanol. And if you don't have any like commercial scientific grade ethanol available, even like vodka works. You can also use formalin, although that inhibits certain testing. So you can't do PCR on samples that are in formalin as easily. So third, collaboration with task force partners. If any of the people working actively on this request, samples or assistance in any regard, if we're able to work with them, that would be awesome. And last but not least is public education. So there's a lot of really good, easy to digest resources out there, such as USGS website, salamanderfungus.org, and we have fact sheets available for these as well that you can feel free to provide to people. So references and any questions.
I don't see any questions, but I want to remind everybody that our communications person, Jennifer, is behind the scenes, and she is putting a lot of the links to the fact sheets and other resources that we've mentioned in the chat boxes, so you can have access to those. All right, thanks, Melissa. We'll turn.
Thanks.
Rachel, next.
So I'm going to talk today about bovine tuberculosis and white-tailed deer. So as you probably know, tuberculosis is a bacterial disease caused by species of Mycobacterium, which is an acid-fast rod. And in the lower right-hand photo, you see dark red rods, which have retained a red dye after being washed with acid. And these are Mycobacteria. Most cases of TB in humans are caused by Mycobacterium tuberculosis. But today I'll be talking about a related species, Mycobacterium bovis, which causes TB in cattle, as well as many species of wildlife and also humans. Bovine tuberculosis in cattle herds has important economic consequences, and so it's monitored by the USDA through skin testing of herds, surveillance of slaughter facilities, and tracking movement of animals between herds. Many species of wildlife can be infected by Mycobacterium bovis. Most of these species are considered spillover hosts. That is, they can become infected, but the pathogen does not persist in the population without recurring introductions from other species. Maintenance hosts, on the other hand, are able to maintain the infection within a population without another source, so they can act as reservoirs of infection for other animals. And the host status of a species, spillover or maintenance, depends on the disease prevalence within a population or area, the resulting pathology within a host, and hence its transmissibility, and also host ecology and behavior. So host status of a species can change from over time or between regions, depending on these various factors. Transmission of bovine tuberculosis from animal to animal is mainly via respiratory secretions, such as you might see in crowded barns or family groups of deer. But it can also be transmitted by the fecal-oral route or by ingestion of contaminated food. The bacteria can survive for months on feed and remain viable for long periods of time in moist and warm soil. Indirect contact at shared feeding sites or stored feed is believed to be the primary transmission route between infected wildlife and cattle, such as at feed or mineral licks that are contaminated with infectious saliva, urine, or feces. Transmission happens more often when animals are in high density, such as at wildlife bait, mineral, or supplemental feeding sites. Transmission of M. bovis to carnivores, such as coyotes, badgers, and raccoons, can occur during scavenging of infected carcasses or by consuming contaminated feed. So maintenance hosts of TB has become a problem in various species around the world, making it very difficult to eradicate the disease in cattle. And these maintenance hosts include the possum in New Zealand, Cape buffalo, warthog, and various species of antelope in southern Africa, red deer and wild boar in Spain, the European badger in Ireland and the UK, feral swine in Hawaii, elk and bison in Canada, and also white-tailed deer in Michigan. And these hosts may carry and shed Mycobacterium bovis for years with no clinical signs, such as in cervids. The disease may progress slowly, as in buffalo, or rapidly, as in possum and badgers. So in North America, we do have bovine tuberculosis in wildlife. And as you can see on this map, it's been identified in 9 geographically distinct wildlife populations and is endemic in the red in 3 populations of cervids, including white-tailed deer in Michigan. Typically, the infection causes chronic progressive disease in cervids, with most infected deer appearing healthy. While some deer may develop severe disease within a few months, many are asymptomatic for years. But over time, the infection can result in gradual debilitation and emaciation. Other common symptoms in cervid surviving to late-stage disease might include weakness, weight loss, coughing, nasal discharge, and difficulty breathing. During field dressing of deer carcasses, hunters may come across lesions associated with TB infection, as you'll see in the pictures at the bottom. And these would include white, yellow, or tan necrotic nodules on the inner surface of the rib cage, on the surface or interior of the lungs, or inside lymph nodes and tonsils. The medial retropharyngeal lymph nodes and the tonsils are the most common sites for lesions in cervids, and usually show signs of infection before other areas. And as Kevin and Beth Buckles mentioned last month in their presentations, pneumonias and thoracic abscesses caused by other organisms can look like TB, so it's important to submit tissues and test for bovine tuberculosis to rule it out. Bovine tuberculosis is a zoonotic disease, so it can be transmitted from animals to humans. Most human cases are associated with consuming unpasteurized milk and dairy products, a problem which is more common in developing countries. But transmission can also occur by direct contact with tissues and fluids of infected animals. This direct transmission from infected deer is important to consider because of the possible exposure to hunters, trappers, processors, and wildlife biologists. And in Michigan, there have been 3 cases of bovine tuberculosis reported that were linked to field dressing infected deer. 2 of these cases were pulmonary TB and most likely came from inhalation of aerosols during removal of infected viscera from deer. The other case was cutaneous, so it was just localized to the skin and probably resulted from a finger injury during the field dressing. So it's important to be aware that it is zoonotic, and use of personal protective equipment such as gloves is important to reduce the risk of contact with the organism as well as other pathogens when working with deer carcasses. So be sure to look for lesions inside the carcass, which might alert you to an infection. Cleaning knives used for field dressing prior to using them to process a deer further is important to reduce the transfer of the bacteria to meat that you might want to eat. Wash your hands thoroughly. And finally, cooking venison to an internal temperature of 165 degrees Fahrenheit will kill Mycobacterium bovis as well as other bacteria. So bovine tuberculosis is currently a big problem in Michigan. In In 1975, the infection was found in a hunter-harvested white-tailed deer. At that time, no one considered it very important, so no actions were taken. Cattle in the state were deemed TB-free in 1979 by the USDA. But then in 1994, bovine TB was found again in a white-tailed deer. This time action was taken and more cases were detected in wild deer. In 1998, the infection was identified in cattle on several farms in the area, presumably due to spillback from infected deer. The disease has become endemic in white-tailed deer in the 4-county area in the northeastern part of Michigan's Lower Peninsula. High deer density and congregation of deer around baiting and supplemental feeding sites, which were widely used in Michigan at the time, probably led to the disease becoming endemic. Baiting and feeding have since been banned since 1998 in the counties where the disease has been found. Increased hunting permits have been used to reduce the deer herd by 50% in the endemic area. However, they still detect the disease at a prevalence rate somewhat below 2%. So it is still out there. In Minnesota, in 2005, bovine TB was discovered in cattle in the northwestern region of the state. They took this seriously because subsequently 2 wild deer associated with these affected farms were found to be infected, which most likely resulted from spillover from infected cattle. So they began surveillance and took aggressive disease management actions designed to eliminate as many deer as possible from this disease management zone around the affected farms. Since surveillance began in deer, 27 infected deer have been discovered, and all of these infected deer were harvested within 16 kilometers of the first infected cattle herd. The last infected deer was killed in November 2009. They've continued sampling of hunter-harvested deer, but that was stopped after the 2012 season, which marked the 3rd consecutive year that no deer tested positive for bovine TB. So their aggressive management effectively eliminated bovine TB in wild deer by reducing the disease incidence to undetectable. So what is the situation in New York? Bovine TB has been reported in wild white-tailed deer in Delaware County back in the 1930s and 1961. In 1992, a TB-positive cow was found on a farm in Steuben County that also raised captive deer and elk. Some of the elk were infected with bovine TB and most likely transmitted it to the cow. Cases were also seen in captive deer herds in 1995 in Steuben County and in 2008 in Columbia County. These outbreaks were managed by depopulating the captive herds. State regulations mandate bovine TB testing for all captive deer herds in New York. And as of August 2020, New York is considered to be free of TB in cattle, but is rated as modified accredited by the USDA for captive cervids, based on a prevalence of less than 0.1%. Feral pigs also exist in New York and along the Pennsylvania border, causing a potential problem in that they can act as a reservoir of disease if the conditions are right. Because Mycobacterium bovis infections in wildlife can exist undetected for years or decades, as was seen in Michigan, surveillance is important, particularly in regions where the bacterium has been historically identified. Currently, the DEC and Cornell Wildlife Health Lab use passive surveillance by testing suspicious lesions in submitted deer carcasses and tissues. In the event that bovine TB is detected in cattle herds, it would be important to shift to more active surveillance to identify and address potential infection in wildlife. So how do we prevent and manage bovine tuberculosis in wildlife? Well, the primary reasons for surveillance and early detection in wildlife include the potential for wildlife species to act as a reservoir for transmission to cattle and the resulting major economic consequences to the cattle industry, the potential for human health impacts, decreased tolerance for infected wild cervid populations on the landscape, and the major expense and aggressive nature of managing the disease once it's established in wildlife. The experience of Michigan and Minnesota have identified strategies to prevent bovine TB from becoming established in wildlife populations, and include rapid response to initial disease detection, adequate surveillance in both cattle and wildlife, and aggressively reducing transmission between animals. Since spread of bovine TB is density dependent, for wild deer, the most effective management option is reduce deer density in a management zone. Minnesota and Michigan have increased hunter harvests of deers, extended hunting seasons, and used sharpshooters to reduce deer density. They've also restricted baiting and supplemental feeding of wildlife to limit congregation of deer at these sites. And for cattle farmers, proper storage of feed to prevent access by deer is important. In Wisconsin, which actually has not seen any TB in free-ranging deer, they screen all lymph node samples collected from deer for CWD testing. They inspect them visually for signs of TB infection, and any abnormal lymph nodes are submitted for culture of the bacteria. Some areas use sentinel species, such as coyotes, which is considered a spillover host for bovine TB surveillance, in that if they become infected by direct contact with the reservoir host, if they see infection within these sentinel surveys, they can assume that infection is present in reservoir hosts. In areas where bovine tuberculosis is established in wildlife, research is ongoing on wildlife vaccines to prevent or lessen the impact and transmission. And there have been field trials in badgers, possums, and African buffalo. So, any questions?
You do have a couple questions. The first one, in long-term infections, once the symptoms subside, how contagious is the animal?
Once the symptoms what?
Subside, once they're no longer symptomatic.
I don't think that they become asymptomatic. I think once they're symptomatic with obvious respiratory signs, they would be shedding the bacterium. And I think probably before they are symptomatic, they can shed in respiratory secretions. If it's like carnivores may get a more intestinal form, and I think they could probably shed it in their feces. Kevin may know more about this. But I think once they're infected, they're potentially transmitting the bacteria.
All right, thank you. And your next question is, what is TB's ability to persist in the environment, or can it only survive inside a host?
No, it can survive in the environment. It can survive, I think, in cattle feces for weeks to months. It can survive in the environment in the right conditions, if it's the right temperature and moisture level, for quite a long time. That's why at supplemental feeding sites, or just in cattle feed that's left out, it may become contaminated by urine and feces and be infectious for quite a long period of time.
All right, thanks, Rachel. So next up, I'm going to talk a little bit about chronic wasting disease, and then I'm going to hand it over to Lauren to talk about how we do chronic wasting disease sampling in New York and some of the other points around sample submission. So share my screen. All right, so I know that I talk to you guys about chronic wasting disease pretty much every year, but there's always new people, and so I think it's important to go over some of the basics with the disease. And from the public, a lot of times I hear, what does CWD look like? Everybody wants to know what they're going to see in a CWD infected deer. And as you've heard today, there are a lot of other deer diseases out there, and we can't really identify them just by looking at the animal. This was one of my research animals that I watched almost every single day, and it died of either CWD or it was scavenged. It was hard to tell, and I would have never ever guessed that this animal had CWD. Similarly, a lot of hunters, shoot a trophy buck and they're really happy about it, and the test results come back and this animal was positive for CWD in Minnesota. So unfortunately, we don't have a really good indicator when the animal is still alive, or even after it passes, of sort of the classic lesions that we've talked about in some of these other diseases. And one of the reasons for that is because the animals don't mount an immune response the same way that they do to some of these other diseases. And that's because the agent that causes CWD is different. It's a prion, or infectious protein particle. So the shape of the prion actually changes, and that's what makes it infectious. And this is in a class of diseases known as transmissible spongiform encephalopathies, or TSEs. These class of diseases are universally fatal. There is no treatment, there is no vaccine, and there is no true genetic resistance. And if an animal gets infected with CWD, it is going to die. We know that deer species, elk, moose, and reindeer are naturally affected by it. And we have seen some recent cases in Scandinavian countries where CWD may occur spontaneously. So the strain of the prion actually occurs differently than what we've seen in North America. But I'll talk a little bit more about the case in Norway. So just some general things about CWD. Once an animal is infected, it's not going to survive as long, and I can show you some data related to that. Infected deer are more likely to be killed by a predator or a hunter. Males or bucks usually have a higher prevalence rate, so they're more likely to get infected than females. And we think this is related to their behaviors. So they have larger home ranges. They tend to interact with a lot of other deer, especially during the rut. And so that may cause them to get infected more frequently. And we have seen population level impacts from CWD. So studies in Wyoming showed that white-tailed deer had 10% annual declines in their population, and mule deer population in heavily infected areas declined at 21% annually. Right now, New York stands out as the only state to have eliminated CWD in free-ranging wild deer once it was detected. Minnesota also did that for a while, but unfortunately, CWD has found its way back into Minnesota. So every time I give a presentation, I have to pull out an updated map. So this map was current as of July. And what I want you to notice is the gray areas are where CWD has been detected in free-ranging populations. The circles represent captive cervid operations. Those that are colored red are where there are animals still in those herds. And one of the reasons for that is because there isn't sufficient funding for indemnity payments to compensate these owners to remove these animals, so to take out that herd. And so in some states, especially Wisconsin, these herds are allowed to continue operation as long as no animals are leaving those herds alive. And so that's something that we do need to work on, because they can continue to be a source of potential infection for wild free-ranging cervids. So the disease has progressed across the country, most likely with human assistance. So as of 2000s, there weren't too many states or provinces that were affected. That increased over the next 10 years. And now there are 23 affected 26 states that have detected CWD at one point or another. You can see New York is still represented on there. So what has worked with CWD? Obviously, New York stands out because of the detection we had in 2005. That occurred in Oneida County, where there were 2 positive captive cervid facilities that found through the herd certification program, found those positives and were able to depopulate those herds. The owner of that index herd was also a taxidermist and a wildlife rehabilitator. And so that kind of set up a perfect storm, but luckily he cooperated. And in one of the areas where he had released animals, there were 2 wild white-tailed deer that were detected with CWD. This was after the DEC set up a very large containment area and did intensive population reduction and sampling within that area for the next 5 years. And there were no subsequent detections. So since then, in New York, we've been using an interagency CWD working group that was put together in 2011. That was especially important because Pennsylvania detected CWD in 2012, both in captive cervids and free-ranging white-tailed deer. Subsequently, the Department of Agriculture and Markets put a ban on all captive cervids, live captive cervids coming into New York State. Prior to that, it had been specific to those states that had had positive detections. Lauren will talk more about our surveillance program using a weighted risk-based system with county point quotas. We have a response plan that is for aggressive action to determine the extent of the outbreak and decrease the localized deer density in that area. And in the last few years, we've been able to finalize and put forth rules around a risk minimization plan that bans the import of intact deer carcasses, doesn't allow feeding statewide, and provides for better education to stakeholders. So one of the things I think is important is to look at the success that different states have had. Illinois is an example of that, where they've used sharpshooting to keep the prevalence rates of CWD relatively low, below 5% in their affected area. But Norway is sort of the more recent one, where they took very extreme action. They discovered a sick reindeer as a result of a captive, or as a result of a collection activity where they were collaring animals with a helicopter. This particular reindeer didn't look quite right. They euthanized it and took it to the lab. It was discovered to have CWD. They subsequently decided to take out that entire herd of reindeer. So over 2,000 animals were culled, and they found 19 total CWD positive animals. And one of the things that they looked at in doing this cull is the success of hunters versus sharpshooters in taking out these animals. And the sharpshooters were much more effective. They were able to use helicopters and snowmobiles, and they culled more animals per day than hunters. So when we're talking about sharpshooting, it's not publicly popular. Hunters don't like it, the public doesn't like it, but from a disease containment perspective, it is the most effective way to go. So if we look at things that have worked, we also want to look at what hasn't worked. When we look at Wisconsin and just the 4 counties where the prevalence rates are the highest, there were 32,000 deer taken out of that area in 1 year. And those went to almost every state in the country. The only state that didn't get any deer from this 4-county area was Delaware. Deer even went to Alaska and Hawaii. So we know that hunters are harvesting animals and likely taking those animals back home, which is so important for us to have that whole carcass ban where they're not bringing in the high risk parts like the brain and spinal cord. We want to make sure that those don't come into the state and then get put out on the landscape where wild deer or scavengers can encounter them. And the reason this is so important is when we look at the prevalence trends in some of these counties in Wisconsin, so this is north central Iowa County for Wisconsin, the prevalence rates for adult bucks in that county are already over 55%. So you have a better than 50-50 shot of getting a trophy animal that has CWD. And it's notable that the prevalence rate in adult does is over 35% now. And some of the modeling work done by researchers in Wisconsin has identified that there may be population impacts if the prevalence in does goes above 27%. You can see that as the epidemic goes on, that the prevalence rates have increased by age class. So what this is showing you is different age categories by the color bars, and then the age classes of adult male, male deer. So the prevalence rates keep increasing by each year this disease event goes on. We also see that survival isn't as good. This is from collared animals. So if an animal is found to be positive, whether it's male or female, the survival rate is considerably reduced, and that goes down quite a bit during the hunting season, which you can see with these dash bars, starting with archery and then very short gun season and even shorter muzzleloader season, where those survival rates decline quite a bit. The other thing that we have to talk about with CWD is that it's not just the deer biologist that's going to be dealing with CWD. Once this hits a state, it's pretty much an all hands on deck event. So this graph demonstrates the staff time for the state of Missouri, their Department of Conservation, in dealing with chronic wasting disease. So they didn't hire 42 new full-time employees to deal with the disease. They were pulling from everywhere within the agency. So people that worked at fish hatcheries were pulling CWD samples because they were sampling 30 to 40,000 deer statewide. So it can impact the agency. One of the common questions is, can humans get CWD? So there have been no known cases of CWD in humans, but we also don't have a lot of people being tested. And would it actually be recognized if it were identified? Would the person get tested? And because of a study done in macaques, which are the species of primate most closely related to humans, where they infected animals with brain material or consumption of skeletal muscle, which is sort of the equivalent of eating venison, they saw that some of these macaques actually came down with a syndrome similar to CWD. Therefore, the Center for Disease Control recommended that nobody knowingly consume CWD positive venison. And this was a pretty big change for them, given that this study still has not been published, but the results were so compelling, they felt it was important to get those out in advance of the actual publication of the study. The reason the study hasn't been published yet is because some of those macaques are still alive, and so they're trying to let them live out their days to complete the study. There are some issues related to other species. There was a study done looking at pigs, where pigs would harbor abnormal prions, and so therefore they didn't get infected, but they might be considered silent carriers. And this concerns me the most because it shows that there might be issues with prion strain adaptation, where these prions are adapting to their host, and that they might become more infectious as they pass through those hosts multiple times. So really what we come down to with CWD is that we've got a public trust issue, where diseased deer are less valuable to the public as a trust resource, because you get issues being told not to consume these animals, or that hunters don't necessarily want to hunt in a CWD positive area. So this puts a financial strain on agencies, not only because you have less revenue from license sales, but also that redirected financial and personnel resources to deal with these CWD outbreaks. And right now there's a really big problem with misinformation around CWD. And I've said this multiple times already on this webinar that we're seeing misinformation around diseases quite a bit. But for CWD, we really need the cooperation of the public as the disease management tool. And if we can't use hunting in the way that we have, we don't have as many options for managing the disease. So the bad news is that we do have misinformation around CWD. We're trying to combat that with some trustworthy sources. The vaccine trials haven't worked. There aren't genetically resistant animals. We will not have a carcasside test in the next 5 years. As nice as it would be to be a hunter and be able to test your animal right in the field, that's not likely to happen. And we have seen CWD spreading in the captive cervid herds despite the USDA programs. But there is some good news. The Association of Fish and Wildlife Agencies in the last few years has put out a best management practices as a roadmap for action for states. And on its own, CWD actually moves relatively slowly. So if we can convince hunters and the public that it's important not to move these products, the disease will slow down. We have some success stories. We don't know of any human cases yet, because that obviously will be a big game changer. And overall, the majority of hunters and the public support CWD management efforts. And so we just really have to overcome those small but vocal minorities. So that's all I have. I would be happy to answer any questions at this time. I see there in one is one in the box. Are there any groups of animals that are noticeably likely and or unlikely to have a disease caused by prions, not just CWD? What other prion diseases are prevalent? So that's a really good question. I didn't have time to get into it, but for TSEs, other notable prion diseases are scrapie in sheep and goats, and bovine spongiform encephalopathy in cows. It was also known as mad cow disease that happened in the UK and other European countries in the 1990s. So that was one that affected humans, and that's really one of the reasons that we're so concerned about CWD, because it is in the same class of diseases, where scrapie in sheep is much more similar to CWD in deer, and there haven't been any known human cases of scrapie, but there are a lot of other human TSEs that are sporadic or genetic. So there's a lot out there. All right, I'm going to hand it over to Lauren at this point.
Thanks, Kristen. So I'm going to talk about the chronic wasting disease surveillance that we have going on in New York, and then kind of some of the sample collection and submission as it relates. So as a general overview of our surveillance program, samples in New York are taken from meat processors and taxidermists through a cooperative program that pays them for their samples. Because of the higher likelihood of detecting CWD in older male deer, we actively sample from taxidermists who we pay more for pulling their own lymph nodes. And this encourages high quality samples. So to improve that likelihood that our sampling will detect CWD positive deer, we use weighted surveillance that combines risk factors such as geographic risks like the Pennsylvania border, the number and location of captive cervid facilities, taxidermist and processor locations, as well as deer density and population. And this allows us to focus our sampling strategies. So what all of this boils down to is a point quota. You'll hear me talk a lot about that in the upcoming season if we work together. These risk factors are combined to give us a targeted point quota for each county that focuses on the quality of samples rather than the quantity of samples. So last year, putting things into a tangible number for New York, we collected 2,817 samples. All the regions surpassed their target goals, as I can show in the graphic. I think that this suggests that there's room to grow. It might be a little bit different this year for COVID. And despite the large number of submissions, only 14 of those were unsuitable samples. They often tend to be salivary glands, which look similar to lymph nodes. So at the end of the presentation, I've added a link to our YouTube video that demonstrates proper lymph node extraction and might be a useful refresher for field labs or folks who are interested. And to encourage the highest quality samples, we send out pre-made kits to taxidermists. That includes the head tag, vial, gloves, scalpel, forceps for easy collection and submission. We sent out over 1,000 of these kits last year. However, a quarter of those weren't returned. I think that this suggests that there are uncertain newcomers or folks that received kits and didn't participate at all, because most taxidermists that have experience with the program return nearly all of their kits. So therefore, I'd like to encourage regional techs and biologists that have direct contacts with taxidermists to regularly check in with their participants, but especially at the beginning of the season. So verify their participation and the number of kits that they need. And then I can always make more and send them directly out to taxidermists as needed. Speaking of those supplies, each regional office is responsible for ordering their own supplies. If you've never used online supply ordering, I've given you the link, and Nick's email, he can set you up with an account And given the current supply chain issues, I think there's going to be extra turnaround time. Please order no later than the end of the month. I did send out a reminder email to folks. We try to be reasonable about how many supplies each region gets. Ultimately, Kevin's in charge of approving the orders. But that just means that we adjust the supplies based on your sampling quotas. And please remember that these are for CWD sampling activities and wildlife health related activities, not field projects. So this means that CWD supplies or supplies related to the submission of specimens to Del Mar. And please try to avoid ordering year-round supplies at this time. We can ship or set aside orders for you, and if you start to run low, we can always get you more. This is kind of what the timeline is looking like, specifically for the taxidermy program. By late August, regional staff should be talking to taxidermists and verifying their participation, like how many kits they need. They might have kits left over from last year. During September, taxidermist kits will be shipped directly by the Wildlife Health Unit to taxidermists. We're going to include the paperwork on that. And we expect regional staff to check in on their taxidermists regularly throughout the season. This maintains a good relationship, make sure that they're doing everything correctly. It also helps avoiding errors. And then during the hunting season, staff will collect the taxidermy kits. Once picked up by the regional staff, please make sure that you're checking for errors such as missing head tag information, missing collector IDs, missing jaws. Taxidermists will receive instructions with their kit and a YouTube video link. Make sure that they have read this information and understand the process, because we had an issue with one taxidermist last year that sent in, 10 kits, which is like $150 worth of stuff, and he missed out on it because it wasn't done correctly. But ultimately, regional staff are responsible for sending the entire kit, including the jaw, to the Wildlife Health Unit. And we can send those additional kits as needed. During the middle of the hunting season, though, I would expect a little bit of a delay. It does take time to get those kits together. So checking for errors and talking with your taxidermist help maintains a relationship that I'm unable to have with your taxidermist. If there is a future problem, you're going to have rapport with them. I won't. And as mentioned in the previous slides, we're requiring the complete taxidermist kits to be sent directly to the Wildlife Health Unit. You can send them in a bag, in a batch, but please do not remove the head tags or the jaws. Don't separate them. You can keep the samples frozen until shipped. We can supply coolers, and this applies even to our furthest lab at John White. Previously, we received the head tags separate from the samples. They were sent out for the purposes of being able to input the points quota and kind of see where the regions were at, but we can adequately keep up with those submissions, so please send them together. Separating those kits really just added confusion and caused serious delays for us. Also send the complete taxidermist kits directly to us. And even if your regional office chooses to age the taxidermist jaws, we appreciate it, but the jaw still needs to be sent to the Wildlife Health Unit for verification. No exception, no jaw, no points. This is what the completed taxidermist kit would look like. We got the head tag and the vial in the smaller bag. The jaw included separately in the larger bag. Do not separate those things, and please tightly secure the caps. We've had problems with them spilling out and contaminating the head tags. Having said that, here's some of the common submission problems that we see. Please make my life a little bit easier. It's my second year doing this, so I'm also trying to learn from trial and error. So I'd like to turn your attention to the collection and submission of heads, now that we've talked to taxidermists. Some of these problems that we've encountered include incomplete, illegible, or bloody head tags, which slows down our data entry, creates errors, causes contamination. Checking the head tags in the field will save everyone, you and myself included, a lot of time. This includes checking for missing collector IDs, which the processor paperwork requires. We're going to discuss that a little bit further in the next few slides. And I can always create and look up collector IDs if you need them. Other problems include improperly packaged heads, kind of like the ones in the photo here. Please use bags for the head, double bag it if you can, put absorbent pads in there. You know, if you kind of wrap the head as you see in the picture and then scoop it up with the bag, it'll be nice and easy, and you'll avoid a contamination for yourself. And then secure the bag with a knot or a zip tie. Those are also things that we can provide in our online submissions. And after you properly package the heads, keep them frozen until you're ready to ship or drop off the heads at the Wildlife Health Unit for processing. As far as the head tags are concerned, once we receive the heads or the head tags, we enter them into our database. The database then calculates the points per sample. While every item on the head tag is important, I've highlighted a few items that I want to talk about. County information is essential to the point process, so no county, no points. And location is important in the event of a positive, as is a hunter phone number, which is a commonly missed field. We also cannot and will not assume the sex of a deer. If it's not filled out, even if you have antler information, you might have an antler doe. Collector ID is assigned and process to each processor or taxidermist, so make sure that this field is filled out when collecting specimens. It's necessary for every head tag, whether or not the processor is receiving payment. And just a reminder that deer that are missing age information cannot and will not be verified, and they might not receive points. And then the last circle that we have on here, some of you might have seen it, these are the newer head tags. This includes the non-random or age biased sampling field to be checked off. You check yes if it's collected by a taxidermist, or if the deer was purposefully collected over others at a busy meat processor based solely on its age. And while all of these items are important for data collection, they're also important to the points quota and ultimately processor payment. While taxidermists get their paperwork with their kits, regional staff are responsible for supplying payment paperwork to their processors. This includes updated or a new standard voucher that I'm going to discuss, and a W-9 that is required only for new processors or for folks who have changed any business information. I'll be sending out an email soon with the new paperwork. Please collect this early on and then submit it to the Wildlife Health Unit as soon as possible. Collecting this paperwork at the end of the season results in serious payment delays. There's some people that need a reminder or 8. And then as a reminder, those standard vouchers are required for anyone that's receiving payment. So this is the updated standard voucher with the new fields that I had mentioned previously. It is required for any participants receiving payment, and regional staff are responsible for getting the vouchers to us. The paperwork can be mailed or emailed directly to us before the end of the season. All the information needs to be exact because minor errors will cause delays or paperwork rejection. This is the state after all. Instructions are on the back, but all the highlighted fields on the form must be filled out. Of the fields, I want to bring attention to a few of those. The payee ID is the social security number or the EIN. Anything other than that is going to get rejected by the state for payment. Payee ID is also not the collector ID. I've added a new box to help avoid some confusion. And again, I can look those up or create one as needed. But the major change is the updated language that clarifies the payment structure that I'm going to talk about on the next slide. So in previous seasons, there have been confusion over the payments. Some collectors thought that all deer were equal pay, and that's not the case. With the standard voucher, including the clarifying language, I thought this might incentivize processors and taxidermists to select the highest quality samples for the highest payment. So to simplify the text, males 2.5 years and older are the highest paid and the highest point value. Females, regardless of age, are 1 point and $5, so on. And then taxidermists get an additional $5 on top of the base pay for pulling their own tissue. Please avoid young deer, and if you didn't pick it up from my previous slide, please double check all the info before you submit it to us. And with that, I welcome you to email me or call me with any questions or concerns, and I'll take questions at this time.
All right, great, Lauren. Thank you. You do have a couple questions. So the first one is, why do the taxidermist samples need to have the jaws with them when the processor samples do not? Can't regional staff age them like they do for all the processor samples?
I know that there's a handful of labs that like to age their own jaws from the processors. It just created confusion for us in trying to streamline the process and kind of get everything on the same page. When it comes to the taxidermist samples, we're also looking at like higher quality samples, older deer. We just want to keep it simple, you know, keep everything together, keep a whole kit together. I think it just would be easier. You're welcome to age them, but we're going to verify them.
Great, thank you. And the last question is, most of the processors' heads are also biased because they keep the fawns out of the collection. Should that bubble be filled in for all heads picked up from a processor or taxidermist?
That's a good point, but we really don't want fawns to begin with. I guess technically it would be considered biased, but no, in that case, you would not fill out that.
Okay. And one more question came in that Jim Stickles is asking, I was told last year that all processors who pull their own samples would not be paid an extra $5, and that offer was mostly just for taxidermists.
So processors, I guess I don't understand the question.
So we pay the taxidermists for pulling their own samples, but we haven't extended that offer to the processors. And I think the reason that we haven't done that is because we can pull the samples from the processor. We were worried about them pulling the wrong tissues. So it's easier just to have the head and pull the tissues directly. And with the taxidermist, because they do their animals one at a time and then discard the rest of the head, it was easier to teach them to pull the lymph nodes directly, and then they can discard the rest of the head.
Yeah, that does make sense.
Right. And there's a couple more comments, just that they can age the deer that we get from the processors, and they do not need to send in the jaw. That's correct.
That's correct. It's just the taxidermist kits.
Mm-hmm. Okay.
Really, with everything being separated in the taxidermist kits last year, you know, between head tag or lymph nodes getting sent separately or no jaw, it just created too much confusion. So I'm just asking for the whole taxidermist kit to come to us.
Absolutely. Great. Well, that's all we have for you all today. I appreciate you sticking with us through all this, and please join for the next virtual wildlife health workshop that we have. It'll be on September 16th at 10 a.m. again. And this workshop will be covering some updates on the New York State Wildlife Health Program and some interesting cases and whatnot. So we hope to see you all then, and we appreciate your time and attention. Thanks, everybody.